several of our students are also at SciLife. Good.
Cool.
Yeah, it's been a heavy day, but we spoke about David will speak about this. David spoke about it several times, so hopefully there's some sort of anticipation effect.
Cool. Okay,
yeah.
so should I
Yeah,
get
please share. We will start in 2 minutes, but you can share so we can just set up.
Yes.
Good, we can see.
Good, good.
Looks nice. Looks well known.
Yeah, you know this picture, you have seen this
yes,
before maybe.
but I think it's very good. This is a lecture that works. So, you know, no, I think it's very important. As you know, we have gone through all sorts of, at this stage, a lot of sample preparation and optics and refraction and all this stuff, quite a lot. So I think this is actually, this is the final lecture about samples that we have. Afterwards, we have a bleaching workshop just after you, but this is the final one. So I think it's sort of the dot on the I. It's really good. This sort of the high end of how to kick out refraction is very welcome.
Okay.
And actually even diffraction. So I still have 1 minute on my computer.
Okay. Yeah. Sure,
yeah.
sure. I'm eager.
It's good. No,
Yeah.
I think we try to stick to time as much as possible. So
yes, sure.
Okay. So I think now it's 45. So shall we get started? I want to welcome David Unre, who has been a speaker here talking about clearing and tissue expansion several years now in our course, and we really like his lecture. So please, David.
Great. Thanks for the introduction. I will just, so now you can see my laser pointer also.
Yes.
Okay, great to be here again. Thanks for inviting me. So I'm a postdoc at KTH at the moment. I'm also affiliated to Karolinska actually, and to the University Clinic of Cologne. And for the past 10 years, I have been working mostly on sample preparation actually. So I started already in 2014 as a PhD student, and already then I started to clear kidney tissue. So this is what I've been doing ever since. And then we have applied these protocols to address different questions about kidney physiology, basically. So yeah, here you see some samples. These are, of course, kidney samples, my favorite samples. So this is an uncleared thick section of a kidney, and this is after you went through a clearing protocol. Here you see also a kidney sample before expansion and after expansion. And as you can see here, expansion protocols are also clearing protocols.
David,
yes.
Sorry, I don't know if everybody has the same issue as me, that suddenly I do not see your presentation. Is everybody like this, or is it just me? I only see your face.
I can see the presentation.
Okay, then keep going, it's just me.
Okay, cool. Yeah, and these protocols are quite sort of, they are related, but I will get back to this in later slides. So I guess you have already gone through all of this. I mean, the problem with biological tissue is that it scatters light. And of course, we are using, when we are performing fluorescence imaging, we are using photons, light. So if they are scattered in the tissue, this creates problems both for the incoming excitation light and the fluorescence photons that leave the tissue to reach your objective and the detector. And again, in the context of the kidney, this is what this problem looks like. So at 10 micrometers, we are okay. At 100 micrometers, we are not so okay anymore. And then at 1 millimeter, we only have noise. So this is, as I'm sure you're all aware of, what we're trying to solve with optical clearing or tissue clearing. And tissue clearing, although it exploded over the past 20 years, and there are now hundreds of different clearing protocols. It's a quite old concept, actually. And there is a German guy called Spalteholz, who he gets the credit for inventing optical clearing. I think this was in 1907, but it was actually invented here in Sweden by Halvar Lundvall in Lund. So he played around with putting human embryos in different solvents. And then he found that a combination of solvents rendered a human embryo transparent. So this is, at least in literature, the first description of optical clearing already, 120 years ago. And what optical clearing is about is to homogenize the refractive index throughout the sample, because this is what is creating the opacity of the tissue. So you have the different constituents of the tissue have different refractive indices, and this scatters photons. So in the interfaces where there's a sudden shift in refractive index, you have scattering of photons. So what we are doing about this is, lipids and proteins, they have different refractive indices, so you can get rid of lipids, since usually we want to study proteins. We can sort of live with getting rid of lipids. We also immerse the tissue in a solution with a high refractive index, because the mean refractive index of tissue is around 1.45 to 1.5. So for instance, in PBS, the tissue is very opaque, but if we immerse it in a solution with higher refractive index, it becomes more transparent. Or you can make sure as much of your sample as possible is of the same substance. So you can use or apply hyperhydration, but I will come back to this in a moment. But all optical clearing protocols, they do one or more of the above to homogenize the refractive index throughout the sample. And this figure is from a review. It's quite old now. It's from 2015, but still, I think this grouping of different clearing protocols, it's the same and it still applies. So the first group is so-called solvent-based clearing, and this is what Halvar Lundvall and Spalteholz was doing already 100 years ago. So this is, usually it consists of two steps. So you have to dehydrate your sample, and this you do using ethanol or tetrahydrofuran, for instance. And then the second step is putting your sample in a clearing solvent. And this is a solvent with a high refractive index. And you see a few examples here to the right. So here so depending on what clearing protocol you apply, you, to different degree, remove lipids from the sample, and you put your sample in some solution or some solvent of a very high refractive index, which matches the environment to the high refractive index of the tissue. So this leaves you with a more transparent sample. Then you have simple immersion protocols, and these are aqueous protocols. And this, I would say, is the most simple version of tissue clearing. So here you just take your sample, you put it in an aqueous solution of a high refractive index. So you see a few examples here to the right, like fructose, for instance, glycerol, sucrose. So So here you're basically doing nothing about the lipid content of the sample. Here you're just matching the refractive index of the environment to be close to the mean refractive index of the tissue. And then again, we have hyperhydration, and here you use urea or other urea-like molecules. And what these molecules do is that they will drive water into the small compartments of the tissue, so that the tissue is now sort of consisting of more water than it was before. And this will also homogenize the refractive index of the tissue. And the last group is hydrogel embedding protocols. And here you introduce a hydrogel to your samples, so you can perfuse with this hydrogel, this monomer solution that you then polymerize. And what you can do after this is then use a harsh detergent, so usually SDS, to remove lipids from the sample. So here, ideally, you want to remove all the lipids from the sample to, in this way, homogenize the refractive index. And then as a last step, you put this tissue hydrogel hybrid in an aqueous solution of high refractive index to still match the refractive index of the tissue to the environment. By the way, if you have any questions as I go, just interrupt me on the way. So as I said, there are now hundreds of different clearing protocols, and it's very hard to say that one is better than the other. It's of course this sort of boring answer that it depends on your application which one to use. Determining factor in my view is usually which protocol is better for your specific staining. So these protocols are slightly different in the way you stain the sample. So for instance, for the hydrogel embedding protocols, you will remove lipids before you stain, which I will get back to later, that this can sometimes be an advantage. Sometimes it's an advantage to use your sort of standard immunoprotocol and just apply it to the non-cleared tissue and then proceed with the clearing protocol, which is the case for most solvent-based protocols. And one other thing I should say is that, of course, these different protocols, they are of different complexity. And usually the less complex they are, like simple immersion protocols, for instance, they render the sample less transparent. But on the other hand, they are simpler. So if your sample isn't very thick, this might be more than sufficient. The larger samples you're working with, the more the higher degree of transparency you need to be able to image them. So then you have to move to more complex protocols. So for our applications, it turned out that aqueous clearing and especially hydrogel-based clearing is what we prefer. But this is mostly, because this works very well with the stainings that we apply. And then, of course, one factor is that, of course, what you are used to working with is usually best for you. So I know all the sort of tweaks, how I can tweak these protocols to work with different kinds of staining. So I'm just used to working with aqueous clearing. And that's why I would use this for my applications. But I'm not sort of taking sides here. All of these protocols have their pros and cons. So again, I will go into a little bit more detail what hydrogel-based optical clearing is about. So I already sort of generally described this, but this is a bit more how it, at least in theory, looks like on the sort of molecular scale. So you have your sample with proteins here in blue and lipids in brown. And then you introduce these acrylamide monomers to the sample together with PFA. So this is just regular PFA fixation. And then you have these acrylamide monomers present that will in turn bind to the PFA molecules. And then as you polymerize the acrylamide, you then have this sort of mesh or this matrix that is attached to all the proteins in your sample. And this is what then allows for removal of lipids with SDS, which is a pretty strong detergent. So then you have removed your lipids with all the proteins sort of still there. And now you can mount your sample in a high refractive index solution, and then you're left with a more transparent sample. And if you want to apply any antibody staining, you would do it after the lipid removal step here. So these are just two examples of kidneys that are cleared. So one here you have T tomato expression in podocytes, which is a cell type in the glomeruli of the kidney. So here you can see this is a quarter of an adult mouse kidney, and you can see all of these filtration units, the glomeruli in the tissue. This is a late embryonic rat kidney, which is stained with the wheat germ agglutinin. And this visualizes the tubules in the kidney, so where the urine travels after filtration in these glomeruli. So both of these samples are around 0.5 centimeter or so in size. And we have, so the curing protocol that I usually use is this one. So this is a simplified version of the original protocol. So in the original protocol, they applied electrophoresis. So they had an electric field over the sample, and in this way they could drive the SDS micelles through the tissue to speed up the clearing. But this has some drawbacks, so no one is using this anymore. So we are doing so-called passive clearing, but the whole protocol is you first fix the sample with sort of standard PFA fixation. Then you do this hydrogel embedding, and as I said, this can be done either in combination, so you can have both the PFA and the acrylamide in one solution, or you can do it sequentially like here. And then you section, if you need to section the sample, you would section it at 200 micrometers to 5 millimeters or so, depending on your research question. You then delipidate your sample. So this is in 4% SDS at 50 degrees. And then you, as a final step, you embed your sample in a high refractive index solution. And here it says STED imaging. So I will get back to this later, but this can of course be any kind of imaging. And what we typically use here for, so this embedding is it's for the STED imaging that we usually apply, but for better transparency, histodens is a better option. So wait, almost. So there have been so about 7-8 years ago, there were some publications showing that you actually don't need to embed your sample in hydrogel. It works just with PFA fixation and then going straight to the delipidation. So this is sort of speaking against everything that I already told you, that you need this sort of matrix, this hydrogel matrix, to keep the morphology of your sample to be able to delipidate with SDS. So this is not really true. So it works also without the hydrogel embedding. The drawback is that for very large samples, you have to delipidate for a long time, and then you might see some compromised morphology at the surface of your sample. So it's just that without the hydrogel, it's like the tissue is less fixed. So it's sort of less resistant to these alterations that you can get from over clearing your sample. So the protocol, this is the protocol that we usually apply today, as at least as a first try, because it's a very, very simple protocol. So we fix the sample, move to sectioning immediately after fixation. We delipidate the sample. So here you can also raise the temperature to up to 70 degrees to speed up the clearing. And then you end up with a much, much shorter protocol, a much simpler protocol. So this is the protocol that I would for if I have a new application or a new research question that I want to answer, then I would start with this protocol. And then increase the complexity if this is needed in the end. So a summary about optical clearing. So as I already said, we mostly use our protocols for high resolution STED microscopy. As I said, our latest protocol is a good sort of starting protocol. It's actually a very, very simple protocol. So it's only fixation of the tissue, delipidation, mounting, and then imaging. But this protocol is not sort of, it doesn't set the record for the transparency. So if you need better transparency, if you want to do whole organs like whole brains, you might want to consider other protocols such as cubic or disco. So I would say that these two are right now the most popular families of clearing protocols. And there are several different versions of these protocols that are compatible with different applications. So if you have reasonably small samples up to maybe 0.5 millimeter up to 1 millimeter, I would say our protocol is a very good starting point. If you are working with several millimeter size samples, then you might want to move to these protocols that are slightly more complex. So yeah, I would always give this advice that start as simple as possible. Even you might not need the delipidation of your sample. So you can just section your sample, stain it, and then try immersion, aqueous immersion clearing. So just immerse your sample in histodens, for instance. If your sample is transparent enough from this for what you need for your application, then you're already good to go. And then increase the complexity just if you need it. So a little bit about staining cleared samples. Of course, life gets simpler if you already have endogenous fluorescence, because staining cleared samples, especially very large samples, can be extremely challenging. But as you all know, of course, you are then more limited with regards to your target. And the clearing protocol has to be compatible, of course, with fluorescent proteins. And some solvent-based protocols are not, so they quench the endogenous fluorescence. You can get around this by then staining for the fluorescent proteins with an antibody, but then you're back to sort of the antibody staining that you have to perform. If you want to stain with antibodies, these probes have to penetrate your thick samples. And this can really be a big issue sometimes. So you need to usually increase the concentration a lot of your antibodies, and I mean a lot. So sometimes up to, if you're working with thin sections and you're using an antibody at like 1 to 500 dilution, you might have to go to 1 to 20 or 1 to 10 even for it to work. And that means that your antibody cannot, it has to be super, super high affinity, because otherwise, at these high concentrations, you will just create a lot of background, and yeah, your contrast is gone, especially at the surface of the sample. Okay, so I think, are there any questions about the clearing protocol, or should we take all the questions at the end, maybe?
Yeah, maybe it's a good idea.
Yeah. Okay, so then we move into tissue expansion. So the idea behind tissue expansion is very different, although these protocols are related to each other. Tissue expansion is about increasing the resolution. So here, instead of working on the microscopy side, as with super resolution microscopy, tissue expansion is to try to circumvent the resolution limit by working on the sample side instead. So the clearing protocol that I just showed you, it's about having this polymer and linking it to the proteins in your sample. And tissue expansion is doing the exact same thing. The difference is that you use these special polymers. So you use sodium acrylate. And sodium acrylate creates these very curled up polymers when you polymerize it in a salt-rich environment. But as soon as you remove the salts from the solution, they straighten up. And this is basically how you achieve expansion of biological tissue. So the first step is exactly the same here. You just have these monomers present that bind to the PFA. But then here in the second step, what you do in almost all tissue expansion protocols is that you immerse your sample in distilled water, and then it will expand. And of course, now these molecules of interest are separated, are more separated in space. So in this way, you have increased your effective resolution. So this doesn't work off the shelf, so to speak. So there is one problem that you have to deal with. And I mean, this is pretty intuitive. If you have biological tissue in your hand, you cannot just pull the tissue apart. It's very, very sort of, you have forces within the tissue that inhibits the expansion of the tissue. And this is the problem that you have to deal with before you can allow this hydrogel to expand. So there are a few different tricks that you can play here. So the first solution, and this was the from the original paper from 2015, is by using oligomer conjugated antibodies. So then you stain your sample as you normally would with an antibody, and you have conjugated these antibodies with these oligomers. And these oligomers you can then link to the gel. And what you can then do is to just use very strong enzymes to just digest away all the protein from your sample. So basically you digest away the whole biological sample itself after you have stained it with the antibodies. So what you are left with is just a fluorescent map of where your target used to be before you digested it away. And now, of course, you only have this gel with the fluorescent molecules in it. And this you can now freely expand in space. The other solution is called ProEXM. It came a year or two later. This is compatible with conventional antibodies. And what you then do is that you just stain with your antibodies that are conjugated to the fluorophore. And then you use different methods to now link these protein fragments in the antibody to the gel. And then you can use, enzymes to digest away parts of your, or at least cut up this antibody. So you just have these small protein fragments that are sort of detached from each other. And by doing so, you can then facilitate the expansion of this gel. The third solution, it's called MAP, Magnified Analysis of the Proteome. Here you don't digest the proteins, but you denature them instead. And this denaturation will then facilitate the expansion of the gel. So here, this is a scheme of how it works. And what they also do in this protocol is to minimize PFA cross-linking. So here they claim that the PFA cross-linking also it creates forces that inhibits the expansion of the sample. So they use a very high acrylamide concentration, and that means that when a PFA molecule binds to an amine group, it immediately finds an acrylamide molecule to bind to instead of cross-linking to other proteins, as you would have normally. Then you polymerize the gel, and after this you denature, and this is very similar to the clearing protocol. So this is used, here you use SDS at a high temperature to denature your sample, to then facilitate the expansion of the sample. And here you reach the same expansion factor as with the other protocols, XM or ProXM. Then there is a fourth solution. This is called cubic X. And here you actually don't introduce any polymer at all. So here they make use of the fact that the tissue itself has hydrogel-like properties. And that means that the tissue can swell and shrink depending on the environment. And they screened a lot of different molecules, and they just found a molecule that swells the sample a factor of 2.5. So this doesn't achieve the same expansion as the other protocol, but still it has the advantage that you don't have to introduce a hydrogel to your sample. And yeah, there are now several protocols out there that all work nicely for different applications. So it's the same as for the clearing protocols, that depending on your application, some protocol is beneficial. So this is sort of a summary of the different families of expansion protocols. So this was the first protocol, XM. This has been shown to work nicely for several applications, but it requires these custom oligoconjugated antibodies, which makes it suboptimal. And I wouldn't say that people are using this protocol anymore. So they moved more towards ProEXM, which works with conventional antibodies, or MAP, which also works with conventional antibodies. So the difference between these two protocols is that for ProEXMs, you stain before you expand your sample, which can sometimes be beneficial. So if you have a staining, for instance, that you know works on with a normal IHC protocol, then you would maybe want to apply this protocol, because then you just prepare your sample as normal and then you proceed with the expansion. For some stainings, the MAP protocol might be better. So here you stain after delipidation. So it's the same as for the hydrogel-based clearing, that you delipidate your sample or denature the sample, and then you stain. And this for some antibodies might be better. And then you have the cubic X protocol, which works with Also with conventional antibodies, here you have no polymers, which means that it's simpler and less toxic, but you have a lower expansion factor, so you compromise your resolution. A few more comments on tissue expansion. So I applied this a lot. So this was in the middle of my PhD work. We use this for so I will show you some results later. We use this for to resolve structures in the kidney. And my impression is that it's very powerful, but it's also very technically quite complex to work with these protocols. So the protocols themselves are often laborious, and especially on the imaging side, it might be challenging. Especially for me, I imaged tissue samples, and one problem you see there is that you always have these uneven parts, so it's very hard to section the tissue completely even. So you will have these bumps on the surface of your tissue, and these bumps will also be expanded. So now you can have quite sort of large parts of your sample far away from the cover slip. And if you then want to image with a high NA objective with a short working distance, this might be an issue to even find places to image. You also mount your sample in DI water, which means that it's a pretty wobbly mounting. So it's prone to drift. This can be an issue. So I would only apply these protocols if you really need the highest resolution for your research question. And if you have access to super resolution microscopes, then this might be a simpler option for some applications. Although this is the selling point of these protocols, that now you don't need a super resolution microscope anymore to achieve super resolution. But I would say in some cases, if you have access to super resolution microscopes, for instance, through a facility, it might be the better option. And also I should mention that there are other expansion protocols, so where you perform iterative expansion. So meaning that you now, with your expanded sample, you introduce another gel, which you then expand, and you can then achieve expansion factors of 20x or even higher than this. But then again, I think this is something, I mean, it's technical enough to work with the normal expansion protocols, and this adds another complexity layer. So it's only for very, very specialized questions, I would say. Okay, so then I will just talk a little bit about the way we have applied these protocols, so my own research. So as I already said, I work with the kidney. So we apply these advanced sample preparation and the super resolution microscopy methods to resolve the kidney filter. And the human kidney is, one of the most important tasks of the human kidney is to filter blood, create urine, and in this way excrete toxins and also tune salt and sugar levels in the blood, or the concentrations of all small molecules basically in the blood. And the filtration, it happens in glomeruli, so we have about 1 million of those in the human body. If we zoom in on the surface of such a capillary, these capillaries are tiny. They are only about 1 red blood cell in diameter, so they are as small as they become in the human body. So if we zoom in on the surface, we find these this very special cell type called podocytes. And podocytes, they make these very, very beautiful sort of protrusions that interdigitate with the protrusions of the neighboring podocytes. And this is a very important part of the filtration barrier. So here we see it in cross-section. This is the blood side. Here's the urine side. You have an endothelial cell layer here, which is also very unique. You have these fenestrations to let through blood plasma for filtration. you have a basement membrane, and then you have this epithelial layer of podocyte food processes up there. And these are tiny structures, so the width of a food process is between 150 and 500 nanometers, and the width of the cell junction here between the food processes is 50 nanometers. So historically, you needed, of course, electron microscopy to visualize these structures. But then we showed in 2015 that we can also resolve these structures using optical microscopy. This we do by staining for proteins that localize to this cell junction, the slit diaphragm, for instance, podosin and nephrin. And what we showed here is that we need to apply optical clearing. So here, as I said before, for some stainings, optical clearing really makes a difference when it comes to the staining quality. And here you see this exemplified. So this is if I stain for nephrin using a normal IHC protocol. This is the result. So I can start resolving these food process structures, but the staining is quite messy. The background is quite high. So we have a not so good contrast. And here I think you can really appreciate the difference it makes with optical clearing. So this is actually the main reason that I apply optical clearing for this application, because here we are not imaging that deep. Of course, it's a bonus also that the sample is more transparent, but primarily it is to increase the staining quality of the sample. Yeah, here again, this is from the paper, just showing the difference here. And this is when staining for podosin. This protein, it localizes to both sides of the cell junction. So it's an intracellular protein located at the slit diaphragm. And here you can see that we can resolve this sort of railway-like pattern of podosin. And this then corresponds to the podosin strands on both sides of the slit diaphragm. And this was then the first time this junction was resolved using light microscopy as opposed to electron microscopy. And together with my collaborators in Cologne, we then started working on how we can quantify changes in food process morphology upon disease. So when you're affected by kidney disease, you have these pathological alterations of the food processes. And this we started to find different ways to quantify. So we developed ImageJ plugins to be able to semi-automatically segment these structures from the images. And this in a mouse model for a type of kidney disease. We then acquired a lot of STED imaging data, and then we applied the analysis protocols, and together with mathematical modeling, we could then publish in 2020 a new sort of theory for how the kidney filter works, and especially why you have leakage of proteins into the urine when you're affected by kidney disease. One thing that we are now working a lot with is to increase the automation, and in this way increase the throughput of these methods. So we are using deep learning based segmentation. So here we are training a deep learning network to segment these structures of interest from the images. And now, so this is a raw STED image, and now we have a very well-trained deep learning network that nicely segments these structures for us. And then, for instance, this image, so with the old semi-automated approaches, this would take several hours to segment all the food processes from an image like this, but now we can do it in just a couple of minutes with the deep learning-based segmentation. So these are very powerful methods. And of course, yeah, they have a wide variety of applications outside of this application. here again is this is what I spoke about before, that we also used tissue expansion to resolve these structures. So this is from a paper in 2018 where we showed that now with tissue expansion we can resolve this sort of railway pattern of podosin in the same way as in a non-expanded sample with the STED microscopy. In this paper, we also showed, so this was the first combination of expansion microscopy with super resolved microscopy. So here we applied STED microscopy to these expanded samples to then even further increase the resolution down to sub 20 nanometers. So summary then of the whole talk is that optical clearing can be applied to prepare samples also for super resolved imaging. So it's not only for large samples. So due to the higher staining quality that you see for some antibodies, it might be beneficial. And also you of course have an improved optical properties of the tissue after clearing. Yeah, and I almost exclusively used clearing plus STED in my research, so I don't use expansion microscopy so much anymore for the reasons that I mentioned before, that these are quite complex protocols. And since we have a STED microscope, I rather work with the STED microscope as opposed to working with expansion protocols. And yeah, there are many new exciting image analysis tools emerging. So this can be really, really helpful, and it can really increase the throughput of your research. So with this, I want to thank you for the attention, and I'm happy to answer any questions.
Thank you, David. So I'm sure there are interested people out there with some questions for their own samples. I think we've had some students, yes? Alejandro, yes.
Okay, I was wondering if the expansion would be more sensitive to different organelles. I'm asking because I work with plants, and most of the water and size control is due to the vacuole volume. So I'm guessing if this kind of expansions can really alter how the cell is distributed inside, just because more water is going to the vacuole or something like that.
Yeah, so this has been shown of course, in the first papers, it was stated that the expansion is completely isotropic, so completely homogeneous in all directions. But this has been proven not true, actually. So there was a paper from a few years ago by the Eggeling group showing that different organelles expand differently. So if you have an expansion factor of your sample of 5, some organelles might expand a factor of 3, some expand a factor of 7. And this is different for all, for different expansion protocols that you apply. So this is something you have to be really careful about. If you want to measure sort of absolute distances in your sample, you have to control that your expansion protocol actually doesn't alter the structure of interest, which yeah, there is a quite high likelihood that it does.
Okay, thanks.
Yeah.
Sadaf?
Yes, thank you. It was very nice lecture. I was wondering if you have ever tested this protocol, like expansion for paraffin embedded sections, and if you know it will work or.
So there are publications showing that it does work. I tried it on paraffin embedded kidney tissue, and the results weren't super nice. But I know I just basically made one try and it didn't work that nicely, so I just dropped it. But there are papers showing that it should work. So I think it depends on the type of sample you have and yeah, what you are staining for and so forth. But in principle, it should work. Yeah.
I have a question as well. Do you know if there are protocols or it's been tried on yeast and bacteria?
I don't know from the top of my head, but I would assume it works. So it's been shown for yeah, almost all types of tissue and cells and organoids, etc. So I think on most kinds of biological material, the principle should work with some tweaks, of course. But I don't know from the top of my head, actually. Yeah.
Yeah, because for yeast, I'm mostly concerned about the impact it would have on the cell wall and how it would actually affect the morphology of the yeast. But that was very interesting. Thank you.
Yeah, that would be, I mean, this has been, of course, a question in all type of tissue. And I think now clearing and also expansion, even though expansion is a little bit more sort of edge, clearing is very accepted, and expansion is also accepted, but it has it's challenging, as you said. But anyway, these two techniques are very well embedded in the community. So I think it's these questions were there from the beginning, and I think they have been sort of cleared.
Yeah.
Sadaf, did you have another question?
Sorry, I forgot to lower hand.
Okay.
Yeah, I can say this also. This was, of course, in the beginning, when these protocols were introduced, this was a big debate. Like, how do they affect the morphology of the tissue, etc., etc. But I think this is one point is that everything you do to your tissue, whether it's fixation, mounting, what mounting media you use, you are affecting the morphology of your sample. So everything is just sort of, yeah, the best representation of reality that you can find, but it's not, nothing is a representation of reality. As soon as you have fixed your sample, you are doing things to it. So yeah.
Yep. So are there any more questions? Okay, well, if not, thank you very much, David, once more.
Thank you. Yeah,
And
thank you so much.
we'll see you next year for sure.
Yeah,
Hopefully.
great. See you next year.
Okay, ciao.
Bye-bye. Ciao.
So I think that we're going to a little bit behind, but we're going to take a 5-minute stretcher because we do need it one, don't we? And then we're going to meet at 3:40 in the other Zoom to continue. Okay, see you guys at 3:40 in the workshop Zoom.
Yeah, see you.